All right, welcome back again, streaming live to all of you, Bio 153 online. And what we're going to look at today is the final part of chapter 28. So it seems like it's a forever-ending saga with chapter 28. It's a very lengthy chapter. And so we're going to be looking at the last part of chapter 28, which deals with sexual development. So that's where we're going to be focusing in on. So, without further ado, here we go. So, when it comes to the matter of determining what the sex will be of the individual, it ultimately comes down to, well, genes, chromosomes, things like that. So we look here at this image, which is looking at the development of the external genitalia of the male and the female. So early on, we have here a time of kind of like a sexual indifference stage. We don't know early on if it's going to be male or female, but eventually, in time, we'll have one sex, either male or female. Ultimately, what will determine the sex of the individual is based off the SRY gene. So the SRY gene will determine the sex, if the individual will be male or female. So SRY is an abbreviation you can see down here, SRY, the sex determining region of the Y chromosome. And hence, when it comes to mom and dad, who gets to determine ultimately the sex of the child, the gender? That will be dad. Dad determines because he has the Y chromosome. So we can kind of see that here. An individual that is male, normally, their chromosomes will be XY. And for a female, the female will be XX. So if that's the case, if the SRY gene is present, yes to the Y chromosome, then we ultimately have male genitalia, which gets formed. If no Y chromosome is present, no SRY gene present, then we have female genitalia, which will be formed. And if you've ever wondered how many chromosomes are found in someone, how many chromosomes is found in a human being? Does anybody know? What is that number of chromosomes, the total number? The total number of chromosomes in a human being, so total chromosomes, and you can see very, very nice, neat handwriting here for you, right? Only the best. Ultimately, the answer would be 46. So 46 is the total amount of chromosomes found in a human being. And how this works is dad will donate 23 chromosomes, while mom will donate 23 chromosomes. Ultimately 46. Some chromosomes are non-sex chromosomes, while others are sex chromosomes. So typically, the majority of them are non-sex chromosomes, which would be the, per se, first 22. And then the last one, 23, then, is the sex chromosome. So that's how that works. A total of 46 chromosomes in a human being. 23 are donated from mom, and 23 chromosomes donated from dad. But what we're going to see is the normal number of chromosomes in human beings is not always the case for everybody. There are some abnormalities found in sex chromosomes, and we look at some of these various syndromes. Take in consideration with females, normally XX for chromosomes. But there are some women in the population that have what's known as Turner syndrome, where they only have one X chromosome and that's it. Some of the symptoms found in the syndrome, Turner syndrome, is usually these women are very short, very, very short. They have undeveloped breast. They are born with no ovaries, wide chest, and they're sterile. And then there are some women who have what's known as the triple X syndrome, XXX. And overall, anatomically speaking, they look like a normal female. But cognitive abilities are affected. They may have learning disabilities and lower than average IQs and even delayed language skills. If they were to take various standardized tests, an IQ test, that would be the case. Okay? So these are just two examples here of abnormalities with sex chromosomes in women, Turner syndrome and the triple X syndrome. Now we take a look at some abnormalities in men with sex chromosomes. We take into consideration that men usually have a chromosome set of XY, but here we see a syndrome known as Klinefelter syndrome. So Klinefelter syndrome, where the man has two X chromosomes, so they are XXY. The symptoms we find here is undeveloped testes, typically. They have very low testosterone levels. They're typically very tall and not a whole lot of muscle mass. They're born sterile because of the undeveloped testicles. Facial hair and body hair do not form, and they also have breast. So they might very well have higher levels of estrogen in comparison to testosterone. Another type of syndrome found in men is the Jacob syndrome, where their chromosome set is XYY. Typically, these men are taller than normal, and they have a below intelligence level. So again, if they were to take a standardized type of test, an IQ test, they would score very low on that. So this is just exposing you to several different conditions that are out there. We're now going to look at how the internal genitalia form. So going back during embryonic development to week 5 to 6 with the embryo, this is considered a time frame of a sexual indifferent stage of development, as we don't know exactly what the overall sex gender will be. What we have early on here for the gonads, they start to develop during the 5th week of development. And what we have early on are what are known as gonadal ridges. Gonadal ridges. So here's a gonadal ridge. And we can go ahead and trace that here for you. Gonadal ridge, gonadal ridge. And within the gonadal ridge is where you have primordial germ cells formed. So these are stem cells, these germ cells, which can become either the spermatogonia or oogonia. Spermatogonia for stem cells for sperm, oogonia for stem cells for eggs, oocytes. Ultimately, the structure here, the gonadal ridge, will develop into 2 possibilities. Gonadal ridge referring to gonads here. So this structure either can develop into the testes or this structure will develop into the ovaries. And that makes sense because, of course, sperm is being developed within the testes. Ovaries is where the eggs are being produced. So the gonadal ridge is what this structure will evolve into. If it's going to be a male, testes, female, ovaries. Other additional structures we see here, we have what's known as the mesonephric duct, also known as the Wolferian duct. And this is referring to male internal genitalia that will form. So this structure, the Wolferian duct, can evolve into a variety of different things. It can develop into the epididymis, the vas deferens, the prostate gland, so a lot of different things like that. With the female, it would be the development of the paramesonephric duct or the Mullerian duct. And this structure will evolve into internal genitalia of the female. It can develop into the fallopian tubes. It can develop into the uterus, the cervix, so things like that. So that's the case here. If the SRY gene is present, male genitalia will form. If the SRY gene is absent, then female genitalia will form. In this picture here, the SRY gene was present, so male genitalia are forming. This would be week 7 to 8 in the embryo of the male. So we can see from the Wolferian duct, the efferent ductules are being formed. The epididymis is forming here. The vas deferens, the seminal vesicle. So a lot of those different types of structures. Of course, the testes is developing from the gonadal ridge, like we mentioned. And then this image here is showing the male what the genitalia would look like at birth. So we can see that here, the vas deferens, the bulbourethra gland, prostate gland, seminal vesicle, epididymis, penis, testis, etc. And then approximately 2 months before birth, testosterone, very critical hormone here, will play a role of stimulation migration of the testicles towards the scrotum, which we'll look at very shortly here. So this picture is showing the overall descent of the testes. Because early on during development, embryonic development, fetal development, the testes are not in the scrotum. They need to descend and move into the scrotum. So early on with development, month 2, 3, 4, etc., the testes start off actually up more towards the kidneys. And slowly but surely, they're making their way closer and closer to the scrotum. What's allowing for them to move, one, is a structure known as the gubernaculum. And the gubernaculum is a very strong fibrous cord that really plays a role with going ahead and moving the testes into the scrotum. Almost like a pulley system is one way to consider it. And then about 2 months before birth, testosterone, very critical hormone in the male, will stimulate the migration of the testicles into the scrotum. Here's month number 7, and then eventually the testicles make their way into the scrotum at birth. There are some men where their testicles never descended into the scrotum, or maybe only one descends. That can be a very big risk factor for possibly the male being sterile. It can increase the chance of the man having cancer. So cancer within the testicles, testicular cancer. So those are big risk factors there. Now taking a look at the female. So we imagine now that the SRY gene is absent, so female genitalia will form. Here we're looking at week 8 to 9 here of the female. So the Mullerian duct, paramesonephric duct, is forming. And that can form the uterine tube or fallopian tube, forming of the uterus here, etc. And of course, the exception here, the ovaries here and here are being formed from the gonadal ridge. Here we can see what the genitalia look like at birth for the female. So we have the ovary, the uterus, uterine tube, the vagina. So gives you an idea. Obviously, these structures are going to grow more, but this gives you a rough idea here. We're now going to take a look and see how the external genitalia form. So we imagine a situation starting at a sexual and different stage of development. So looking at the development of the homologous structures, and that's referring to the external genitalia. So early on, these structures are color-coded, and here they are in the bold in the black. So we have the urethra fold, urethra groove, the genital tubercle, and labioscrotal swelling. If the case the SRY gene is present, then male genitalia will form, and the urethra fold will evolve into the spongy urethra found within the penis. The urethra groove will evolve into the man's urogenital sinus. So we can't exactly see that here, but we will on the next slide. The genital tubercle will develop into the man's penis, especially the glans penis, and the labioscrotal swelling in yellow will develop into the man's scrotum. Here we can see approximately at 5 weeks of development, and we can see the development of these structures here, the glans penis in the blue, penis, scrotum in yellow. We can see the, well, we can't see the spongy urethra, of course, but the urogenital sinus would be in the pink here. So I'll highlight that here for you. So right here, this is the urogenital sinus right there. Women don't have that structure. Goes along the middle median area of the penis through the scrotum towards the man's anus. Now we imagine that the homologous structures evolved into female genitalia because that SRY gene is absent. So the urethra fold, in this case, evolves into the labia minora, the inner skin fold of the woman around the vagina. The genital tubercle will evolve into the clitoris instead of the penis, and the labioscrotal swelling will develop into the labia majora. So that's the outer skin fold when we consider that around the vagina. And then the urethra groove will develop into what's known as the vestibule, and this is kind of a regional area that contains the opening of the woman's vagina and the opening of her urethra orifice where she'll urinate. Here we can see the development of these structures, approximately 5 weeks here. Ultimately, kind of more what items look like at birth. So the clitoris there in the blue, the labia majora in the yellow there, and the labia minora in the pink. And again, the vestibule, we can't exactly see that per se. It's not signaled here, but it would be this opening here, this regional area here that contains, again, the opening of the vagina and the opening of the urethral orifice for urination purposes. So very important to make sure to know these homologous structures at the sexual and different stage of development, and to know what do these structures evolve into when in regards to the gender. Do these homologous structures, do they evolve into male structures, female structures? So important to know the equivalencies, very important to know. All right, and now we have a couple review questions to see if we're picking up on this very important information. So our first one here today, number 7. So during sexual development of the external genitalia in a male, the genital tubercle evolves into the what? So what would be the correct answer for this one? And the correct answer here would be choice letter A, the penis. So the penis would be the correct answer here, the penis. All right, number 8. During sexual development of the external genitalia in the female, the labioscrotal swelling evolves into what? So what would be the correct answer for this question? And the correct answer here, I would go with choice letter D, the labia majora. The labia majora. Number 9, a male that has undeveloped testes, breast development, and is sterile would have what syndrome? So what would be the correct answer here? And the correct answer here, I would probably go with choice letter C, Klinefelter syndrome. So that would be the correct answer for this question here. All right. So I hope you enjoyed today's lecture. Oh, and you may want to also just maybe briefly review these tables I have at the end here. So, all right, signing off.